All right. Hello, everybody, and welcome back to some more biotechnology slides. It's Wednesday today, and Friday is, I know I think I put it on I put it on some announcement somewhere, but Friday is the end of the unit and when our unit test is going to be. So, over all of these different types of biotechnology, I will have a review for you. I have a Quizlet made to help you review that. Maybe I should put that on for today. Hopefully, I'll remember to do that. Anyway, so this type of biotechnology, which actually you might be hearing a little bit in the news about COVID-19 testing because they're using PCR to help do that. Anyway, so PCR stands for polymerase chain reaction, and it's this reaction that was developed by, I don't know who it was developed by. I can't remember off the top of my head. But it was developed by this person, and what's really, really awesome about it is that it can take a really teeny, teeny, tiny, small amount of DNA, and then you run the PCR on it, and it amplifies it instead. And then you will have millions and millions and millions of copies of that DNA. And so then there's really never like a question, or like you're not worried like, oh my gosh, are we going to run out of DNA? And that will come into play for certain types of jobs and professions to where they need that. Anyway, so it says at times you need more DNA than you have. So, like at a crime scene, for instance, they may find a little bit of DNA, but what if they only have like such a small amount and they can barely even run like one test on it? PCR solves that problem. You run a PCR on that DNA found on the crime scene. Now you basically have an unlimited amount of DNA that was found at the crime scene that you can keep running tests on. And so that's why, in law enforcement and figuring, out how or trying to solve crimes, PCR is hugely useful. It gets lots of copies of the gene. PCR, it can make millions of pieces of DNA, copies of DNA out of just a small, small amount. This is like a picture that goes through how it all works. You don't need to know about the denature anneal primers and all that kind of stuff. For PCR, for me and COVID-19, as long as you know that it can take a small amount of DNA and make millions and millions of copies of it, you're good. This copies of DNA, it amplifies it, and it's similar to DNA replication. I don't know if when we did our molecular genetics unit and learned about the structure of DNA and protein synthesis, one of the things that we also did learn about was DNA replication. As a reminder, what does that mean? DNA is DNA. Replication means to make a copy of it, and basically that's exactly what PCR is doing. This goes through like all the materials and things that you need for it. Again, like I said, I'm not as concerned that you don't need to know all of these steps. It's just there more for your information. But as it does relate to, I'm going to move myself down over here. As it does relate to COVID-19 and testing, anybody get tested out there? They did have it like the last couple of weeks at the Rush Center that you could go get tested for free. And I was like so tempted to go do it. I just wanted to just see. Not that, I'm not sick or anything, and I don't think I have it, but I just wanted to see, but I didn't go. Anyway, healthcare workers, they are using PCR. There's a couple of companies that developed a test using PCR to tell if somebody has COVID-19 or not. And it's a really, really quick process. Like when we were done with school, like what was it, March 16th, that was our last day. Then St. Patty's Day, the 17th, we didn't have school. It was taking a week or more to be able to get tested for COVID-19. Thankfully, in the last couple of months, that definitely the process has been made more efficient, and it's been made more efficient through PCR. It can detect even very small amounts of the virus by duplicating it and then running their tests on it. So, one of the reasons why I didn't go get my COVID-19 test is because they literally have to stick this big, huge, long Q-tip like up into your nose and kind of like it'll scrape the back of your throat. And I was just kind of like, ugh. But I just wanted to. I should have just for the sake of science. Anyway, and then that will get, if there's any viral particles or virus in there, that's where it would be. And then they run it through this PCR that they have set up just to test for for COVID-19. And yes, okay, wait, shoot, come on, pause. Okay, I think we're back again. Sorry, somebody just came to the door and I had to go get it. But anyway, we are using PCR to help um, get the results back from the tests of if you have COVID-19 or not, get it back faster. So it's obviously a huge use right now and in the news. I don't know if you've, maybe the next time you're listening to that or on the news or something, see if you hear anything about PCR and testing. Okay, stem cells. Switching gears to another type of biotechnology. I think you probably have heard of stem cells before. Actually, no, this isn't recording. Is this recording? I don't know. Oh, it is recording, but I don't know if it's seeing me. Camera. Oh, there I am. Sorry. Ugh, thrown off my game here. Stem cells. You probably have heard of them before. Stem cells are really, really, really cool. It says right here an undifferentiated cell of a multicellular organism that is capable of giving rise to indefinitely more cells of the same type, and from which certain other kinds of cells arise by differentiation. Whoa, what the heck does that even mean? Basically, it means that a stem cell has the ability to turn into any type of cell. We all started out sperm and egg met and the cell started dividing. We were all stem cells at once. You still do have stem cells in you. So there are a couple different types of stem cells. Embryonic stem cells, they are derived, come from embryos. Again, remember when I said we were all stem cells as when we were an embryo. Um, so are from embryos at a developmental stage before the time of implantation would occur in the uterus. So sperm and egg met, conception happen, the cells start dividing for probably like about 3 to 4 days, maybe 5, before that actual embryo implants into the woman's uterus. It's an embryonic stem cell, or that's where those are embryonic stem cells. What's really controversial about them and using embryonic stem cells is that are you ending a life? And so, embryonic stem cell research in the United States, uh, you're not really able to do it because there's such a controversy about is that ending a life or not. So, but that's what an embryonic stem cell is. We think that those have the best potential to turn into any type of cell. It could be, nerve cells, blood cells, muscle cells, bone cells, whatever type of cell. Okay. Another is called are called embryonic or adult stem cells or somatic stem cells. That's what you have in you right now. Now, is it because you're not technically an adult right now? So, do you have teenage stem cells? No, they are like adult stem cells. They are undifferentiated, found among differentiated cells in a tissue or organ. They can renew themselves and can differentiate to yield some of all the major specialized cells, but typically, in your blood, like the blood vessels, it's going to give rise to blood cells. Um, in your muscle cells, it's going to give rise to more muscle cells. It's not like, your heart muscle cells, they're going to develop into a brain cell. That's not going to happen. So they're a little bit more limited on what type of cell that they can turn into. But we do have them. If you've ever heard of anybody getting a stem cell transplant, they usually hopefully can take their own stem cells. It's a treatment used for cancer patients a lot of times, um, and it can be used as a treatment for that. But now, since technology has moved along and gets even better in medicine, there's this other type called induced pluripotent stem cells. They are derived from skin or blood cells and have been reprogrammed into an embryonic like state that enables them to then turn into any type of cell. So what's great about these is that you don't have to like use an embryo and a life, depending on whatever you think or you believe. You don't have to do that. You could take your own skin cells, they can reprogram them back into that embryonic state, which is really, really freaking cool that we can do something like that. And then here is everybody's favorite, the Amoeba Sisters. Watch the video, and then I do have a formative. And like I said, I probably am going to put the Quizlet on today's topic just so you can start studying. Hope I should probably put the review too. Okay, I hope this video isn't like too messed up because of like me pausing and then the video, the my camera wasn't on. We'll just see what happens. And hopefully you guys laugh at me or something. Okay, bye y'all.